Welcome to this video on biology revision with revising biology paper 1. The fourth topic is pretty small, bioenergetics, so basically photosynthesis, respiration and metabolism. All fairly easy topics but all vital to understanding a lot of biology. So without further ado, we're going to look at what's going to be coming up in your exams. So if you're taking Combined Science Foundation, your main focus of this video are the questions on photosynthesis and the photosynthesis required practical, which will both be marked out in the video. And you do not need to know anything on respiration or metabolism, that's been all taken out. And the last two questions on photosynthesis about the uses of glucose, they've been taken out as well, so don't worry about them. If you are Combined Science higher, then again your major focus, photosynthesis and the photosynthesis practical. But what's been taken out of yours, once again, the uses of glucose from photosynthesis, but this time as well, the response to exercise bit, this has actually been taken out of everyone's exam, so I don't have any questions. We're not going to be going over it at all because it's not going to be in. So you guys, it's just important that you skip the final two questions on photosynthesis, but all of the rest of it is important for you to do. So if you're taking biology as a separate science, or triple science as I refer to it as, once again, your major focus, photosynthesis, the photosynthesis required practical, what's been taken out of your exam, this is basically all of the respiration stuff. So metabolism, and this is the respiration stuff. You don't need to worry about that, as well as the uses of glucose that's been taken out of your exam as well. And a triple science higher, you don't actually have a major focus, but everything in here might come up, apart from the response to exercise that we're not going to go over, and the required practical. So we're going to start with these 12 questions on photosynthesis, so pause the video and have a go, and don't forget to miss out the questions that aren't going to be coming up in your exam. So you need to know the word and symbol equation for photosynthesis inside out. Here is the balanced symbol equation. So you've got carbon dioxide, CO2, oxygen, not oxygen, water here, H2O. Glucose is one you probably won't know, and you need to remember this. Glucose is C6H12O6, and oxygen is O2. It's diatomic, goes around in pairs. We cover that in chemistry. And to balance it, you have 6 CO2, 6 H2O, and 6 O2. So it's kind of the devil's equation because you've got 6, 6, 6, but no 6 on glucose. And the word equation, obviously, carbon dioxide plus water leads to glucose plus oxygen. Photosynthesis is an endothermic reaction. So what that means is energy is being taken in from the surrounding environment to form the products. So sometimes you will see light written above the arrow, and this means that the reaction is taking in light energy. Of course, we've studied how chloroplasts carry out photosynthesis. They have chlorophyll, that green pigment which absorbs sunlight. That's why they need sunlight to provide the energy for this process to happen. So there are 4 factors affecting the rate of photosynthesis, and the next whole bit is essentially built around this. It's really important you are familiar with these 4 factors. So the first one is light intensity, or the amount of light the plant's getting, because obviously the plant needs enough light to absorb that light energy for photosynthesis. So if it's not getting enough light, then it won't be able to do enough photosynthesis. The second one is carbon dioxide, because that's one of our reactants in the photosynthesis equation, and obviously if there's not enough carbon dioxide, then the reaction won't happen as fast. And the third one is temperature, which is interesting to remember. It doesn't have anything to do with the reactants or anything needed this time. Temperature is needed because of the enzymes that catalyze photosynthesis. They need the optimum temperature to work at their optimum pace. If it's too hot, they'll get denatured and photosynthesis can't happen. And the fourth factor is the amount of chlorophyll in the plant. And this is obviously important because the chlorophyll absorbs the light needed for photosynthesis, and the amount of chlorophyll could be affected by a disease or a lack of nutrients. But yeah, chlorophyll are also very important to photosynthesis. So here are the graphs for the factors that affect the rate of photosynthesis. We have light intensity, we have temperature. CO2 is exactly the same as light intensity, so this could also be the carbon dioxide graph, and we don't really need to worry about the graph for chlorophyll. So what the light intensity or carbon dioxide graph shows is that as we increase the light intensity, the rate of photosynthesis increases, but only up until a point, and then it flattens out. You can increase the light intensity you want, but the rate of photosynthesis isn't going to increase anymore. And what happens here is we say that light intensity is no longer the limiting factor of photosynthesis, and this means that there's another factor that's limiting the rate of photosynthesis, stopping that rate from increasing any further. So it could be that it's too cold for the enzymes to physically react any faster, even though you have all the light energy you need for it. Or it could be carbon dioxide. This is exactly the same as the carbon dioxide graph. It could be that you have all the carbon dioxide that you need, but you might need a bit more light for that energy to allow the reaction to happen. With temperature, it's a bit different. As we increase the temperature, it increases, but we reach a peak at around 37 degrees C. This is because it's where the enzymes have their optimum temperature. They're working fastest. But then as we increase just a little bit more, we go too far. It could only be about 10 degrees or so. All the enzymes denature because it's too hot for them. It's not in their optimum range, and no photosynthesis can happen at all because there's no enzymes to make it happen. So a graph that shows more than one factor might look like this. So we have a graph showing both the effect of temperature and light intensity here. As you can see, maybe there's been two experiments, one at 25 and one at 20. As we can see, as we increase the light intensity at both, the rate of photosynthesis increases. But then at 20, it starts to flatten out. And this shows that 20 degrees C temperature must have been a limiting factor, because no matter how much we increase the light intensity from there on, we didn't get any more rate increase. However, when we increased the temperature by 5 degrees, our light intensity carries on increasing the rate of photosynthesis. So light intensity is still the limiting factor, but then at this point here, or it would have been at about, I don't know, this point here on the graph, as soon as it goes flat, basically, ignore this, it's just paint being weird. I can't make it completely flat. I don't have a, well, I could have used a line, but pretend this is completely flat. But yeah, here is where light intensity no longer becomes the limiting factor. Temperature, or at the top one, it could have been CO2, we don't know, is the new limiting factor. And yeah, just make sure you're able to read these graphs that show multiple factors and understand what's going on, because you may well see them, especially if you're in the higher tier or doing a higher tier exam. So if you've been watching the bit with graphs, you should now be familiar with what a limiting factor is. This is only really if you're taking a higher exam. If you're taking foundation, make sure you're aware of the graph shapes, but chances are you're not going to be assessed on what a limiting factor is. It's essentially the factor that limits the rate of photosynthesis. It's really not that hard or complicated to get your head around. You also need to know about how we can use those factors that affect the rate of photosynthesis to enhance the growth of plants, and how this can benefit farmers or plant growers that want to increase the yield of crops that they get. So say a farmer knows that light intensity, let's take light intensity, is their limiting factor. Chances are in the day it's not going to be because they'll have the sun, but maybe in the evening they want to install an artificial light so that the plants can photosynthesize for longer, grow more, and produce, again, a bigger yield. Maybe CO2, carbon dioxide, is the limiting factor. Maybe they've carried out a study to find that out. So a clever way of increasing the level of CO2 in the greenhouse is by using a paraffin heater, which increases the heat, But by burning paraffin, it also produces carbon dioxide because it's a type of oil. If they know that temperature is the limiting factor, then again they can use a heater such as a paraffin heater, but in the summer it may get too hot, and this means that they might have to open a window, introduce ventilation, or give the plants some shade to stop the enzymes from denaturing. And of course it's worth mentioning that greenhouses themselves are built to make plants warmer by allowing all the sun to shine through. Just be aware that the farmer needs to balance out increasing the abundance of these factors, increasing the abundance of temperature, etc., what we've just discussed, with the cost, because if they're going to spend loads and loads of money to increase the yield, but then their extra yield doesn't earn them as much money back, then it's not worth doing these things in the first place. So just remember, you could be asked a question, is it worth the farmer doing it? You might have to calculate the cost, you might have to calculate the increased yield. It's all about balancing up growing a higher yield and the cost of growing this higher yield and getting that balance just right. So now we're going to go through the required practical of this section. It's an experiment to investigate the effect of light intensity on the rate of photosynthesis. So as we know, light intensity increases the rate of photosynthesis as it gets brighter. So the way we're going to set this up is we're going to use pondweed in water. There's two ways of setting it up. There's the easy way and there's the scientifically accurate way. The easy way is you can just place your pondweed in a beaker of water, and as you carry out the experiment, you can count the amount of bubbles produced by the pondweed, as that's the oxygen produced from the photosynthesis leaving the plant. So the more oxygen there is, the more photosynthesis is happening. So you can tell the rate from that. Or if you want to, you can do it in a test tube, bung the top of the test tube and feed a water-filled capillary tube to a syringe, and then as the oxygen comes up, you can fill the syringe up. But I'm sure counting the bubbles should be fine in a method if you get asked it in the exam. So what you're going to do is, when you've got that beaker, you've got your pondweed in with water, you're going to take a ruler, lie it along, and at a specific distance, maybe start with 10 centimeters, you're going to place a light source. So that might be a lamp. You're going to turn it on, shine it into the pondweed, and you need to allow the pondweed to acclimatize to its new conditions and get that rate up to the speed using the new light. So leave it to stand for a couple of minutes, and then you're going to time yourself for a minute and count all the bubbles that are produced, and then you can record that. You can move the light source back from 10 to 20 centimeters, move it back 10 centimeters. You again leave it to acclimatize for a few minutes, and then again count the bubbles, and then you can repeat, moving the light source back, leaving it to acclimatize, counting the bubbles until you reach a meter, or however long you've chosen to do it. Obviously, if you're doing it with the syringe, you can measure the volume of gas you've collected in your syringe, and then if you want to be scientifically accurate, you can repeat it 3 more times. Add that in if you've got an exam question which has described the method. That's very important, and I think that's probably got you covered for the required practical. So there's a slightly complicated law to calculate how the light intensity and the distance the lamp is away from the plant is related. It's called the inverse square law, and you should be kind of familiar with inverse and direct proportion from maths. Well, this is inverse proportion. So the light intensity is directly proportional to 1 divided by the distance squared. That is the inverse square law. And so how it works is, if you halve the distance, the light intensity will be 4 times greater, for example. And if you increase the light intensity by 9, for example, the distance will be thirded, if that makes any sense. So I'm going to trust that you're familiar with inverse proportion from maths. Just the important thing that you need to remember that's not from maths is light intensity is 1 divided by the distance squared. So you also need to know about the useful product, glucose, how the plant uses it, because obviously it wouldn't be photosynthesizing if all the products were useless, it needs glucose. There's 5 ways you need to know about. The first one is glucose is used in respiration. This releases energy. We're going to look at respiration in just a second. It releases energy to allow the plant to, use energy, do various bits and pieces. The second way is the glucose can be stored as starch. Glucose is better stored as starch because the starch is insoluble. You imagine the glucose is a sugar, so it's a simple carbohydrate. Starch is a more complex carbohydrate. So, again, starch is insoluble. That's why plants store glucose as starch. Glucose can also be made into cellulose, and that's used to make the plant cell walls to protect the plant cells. If you remember right back to the beginning of these videos, glucose is also combined with nitrate ions to make amino acids, which then go on to make proteins. And glucose can also be stored as lipids, so it can be turned into oils or fats, and this can allow it to be stored in seeds. Plants need glucose and nitrate ions to make proteins, so the glucose is combined with the nitrate ions to make amino acids. The amino acids we look at this in paper 2, the amino acids go on to make proteins. So here are 12 respiration and metabolism questions if you want to pause and have a go. Respiration is a bit of a tricky one to get your head around when you first hear it, because for some reason everyone hears the word respiration and they think of breathing, but that's not it at all. So we say respiration goes on in the mitochondria, and well, we covered that in the first video, didn't we, with subcellular structures. When it happens all the time, photosynthesis in plants only happens during the day. Respiration happens during the day and night in plants, and it also happens in humans and all animals, all living things. And what respiration is, what is it? Well, it's basically transferring the energy from glucose. So it's the reverse of photosynthesis. We're going to look at the whether it's exo or endothermic. It is exothermic because you are essentially oxidizing that glucose. So you're reacting glucose with oxygen, the two products of photosynthesis. Your body doesn't do photosynthesis, so you've got glucose and oxygen, you put it together, and you get out carbon dioxide and water. So although photosynthesis does not happen in humans, respiration does, and it's pretty much the reverse. And what happens is that process of oxidizing glucose, that releases the energy from glucose. That's what you need to know. It's exothermic because it releases energy into the surrounding environment. As I told you, respiration is the reverse of photosynthesis. So what you've got, you expect the word and balance symbol equation to be the same, well, obviously switched around. So you've got this glucose, remember the C6H12O6 with 6 O2s reacts to make 6 CO2, 6 carbon dioxides, 6 H2O, 6 water, and energy, lots of energy produced from glucose. Now, I should have mentioned what we've just been talking about is aerobic respiration. Now, aerobic, you're going to see it come up a couple of times in biology, more in paper 2. It simply means with oxygen. So then when we go on to question 4 and talk about anaerobic, this means without oxygen. So there's 2 types of respiration that can happen in the body. We're about to look at the anaerobic respiration equation. So anaerobic respiration is, well, let's think about aerobic respiration. Glucose plus oxygen makes carbon dioxide plus water. If we take away the oxygen because it's anaerobic respiration, we simply get glucose and it gets broken down into lactic acid. So anaerobic respiration in animals, it goes from glucose to lactic acid. It's the incomplete breakdown of glucose. And we would talk about how this is removed and all that, but that's been cut out of the exam, so we don't need to. In plants, again, it's a bit different. It's just glucose on its own, but it gets broken down not into lactic acid, but into ethanol and carbon dioxide, which we're going to look at later. So the differences between aerobic and anaerobic respiration, there's 3. First one, it could not be more obvious. Aerobic uses oxygen. Anaerobic does not use oxygen. Aerobic respiration produces more energy because you're oxidizing it. The glucose is being fully oxidized and completely broken down, whereas in anaerobic respiration you get this incomplete breakdown and not nearly as much energy is produced. Anaerobic respiration only happens when we can't get enough oxygen around our body quick enough, so when we exercise, but again that bit has been cut out of the exam for everyone. And the final difference, you could talk about the difference in products. Aerobic respiration produces carbon dioxide and water. Anaerobic respiration just produces lactic acid. And you may have heard about it talked about, especially if you've done PE GCSE, you will have heard lactic acid building up in your muscles being talked about. But if you haven't, don't really worry. And just as we looked at what the glucose produced from photosynthesis is used for in plants, we're going to look at why we as animals need that energy from glucose. What we need it for, well, first of all, movement. We need to move, contracting our muscles, doing exercise. We also need it to keep warm. That energy is used to be can be used to heat us, and we can also use it to build up larger molecules in chemical reactions. Again, we're going to look at this in metabolism down here. So anaerobic respiration, we've been over this already pretty much. Where does it happen? In the muscles. When does it happen? When we can't get enough oxygen to them. Again, that kind of bit has almost been cut out, but I'm putting it in there anyway, just in case, you never know. So, as I told you, we can use anaerobic respiration in industry using the plant one, because lactic acid is no good to anyone. What do plants produce in anaerobic respiration? And yeast as well, remember. Ethanol and carbon dioxide, I said, didn't I? So we can use both of those products. So if you think about it, we add yeast to bread, don't we? And the reason, because if you did food tech, you'll know why this carbon dioxide, when you put the bread in the oven, the yeast produces all these carbon dioxide bubbles, and they bubble up, and it's a raising agent. It makes the makes the bread rise. And when yeast carries out anaerobic respiration, we call it fermentation. You may have heard that name used for different foods. We also use fermentation in the alcohol industry, because ethanol, obviously we use it to go on and make alcohol. So especially beer, we use the fermentation of yeast to make beer and wine, because it produces the alcohol that then goes in that beer and wine. So what is our metabolism? Metabolism essentially describes all the chemical reactions going on in our body. And the majority of the chemical reactions going on in our body are either our gut breaking down our proteins, carbs and fats that we consume. They go down, they get broken up. Do you remember what they get broken up into? Maybe you need to go and check out biology, the second topic, cell organization. We cover that. If you can't remember that, that's very important, coming up in your exam in a week. And then these small products that our body takes them, and we go and then make new proteins, carbs and lipids to perform functions in our body, and those are the majority of the chemical reactions that go on in our body. Metabolism again describes all of them. What catalyzes chemical reactions in the body? This should be the easiest question on this PowerPoint by far. Enzymes. If you don't know that, you should go back and check out cell organization. We have a whole thing on enzymes and digestion, very important. What does the body use sugar, amino acids, fatty acids and glycerol for? Again, I literally just talked about that. These are, sugar, we break down complex carbohydrates into sugar, which is a simple carbohydrate. Proteins get broken down into amino acids, and lipids, fats and oils, get broken down into 3 fatty acids and 1 glycerol. And then we can use this sugar to make new carbohydrates for our body to use. We can use the amino acids to make new proteins for our bodies to use. Again, we cover this in paper 2. It's important you have protein synthesis goes on in your ribosomes. We study that in the first topic of paper 1. You need to know that for this exam. And the fatty acids and glycerol can be used to make new fats, for energy storage, for whatever. You don't really need to worry about that. Okay, so examples of chemical reactions. Again, we talked about it. This is either large molecules being broken down into these guys, or these guys making up the proteins, carbs, and fats. So, for example, our sugar molecules, remember glucose is sugar, so our glucose molecules, they could be joined together to form starch or glycogen. Glycogen is essentially what we use to store sugar and energy in us. So like plants use starch to store their glucose, we use glycogen to store our glucose. That's quite important. Again, lipid molecules we can make from fatty acids and glycerols. We can make fats and oils from them. We use our glucose, which is a type of sugar, in respiration to release energy for the body. And protein gets broken down into urea to be excreted in urine. So those are the chemical reactions that happen in the body, really. Most of them are covered at some other point in the specification, but this is kind of collection because we're studying metabolism, the total of all the chemical reactions that happen in our body. And there we are finished with, well, everything for paper 1. If you've watched all 4 biology videos, which I strongly recommend you do if you want to get an amazing grade in 8 days' time. Remember, exam questions, practice exam questions, once you've been through all the questions and all the PowerPoints, you're happy that you know 99% of the stuff that you need to know, because remember, you don't need to know all of it. But bits have been taken out of all your exams. As you can see here, there's bits taken out of everyone's exams just in this small final unit. So remember, exam practice questions, do these, come back to the ones that you get wrong. Maybe the night before you want to come back to all of them, have another look over them, make sure you know them all. That will be very important as well. But for now, I hope you found this useful. I hope if you have found this useful that you'll subscribe so that other people can find the channel more easily. And I hope you get an amazing grade in your GCSE.